Have you ever heard of sickle cell anemia, Alzheimer's disease, or cancer? All of these diseases are caused by a change in DNA, which ultimately leads to a change in a protein that causes it to malfunction. Here we discuss these seemingly insignificant DNA changes or mutations that can cause absolutely catastrophic effects on the body. Most cells in a multicellular organism contain the same set of genes. Some genes essential for life are expressed, meaning transcribed and translated, in all cells. An example would be the cellular respiration genes that encode the enzymes that are involved in the metabolic pathways needed to make ATP. Other genes are expressed only in certain cells to give cells specialized characteristics. For example, only pancreas cells synthesize insulin, which is a hormone released after eating, to signal to your cells that blood sugar is high. What this means is that while many cells contain the gene that encodes for the insulin protein hormone, only pancreas cells actually express this gene. Gene expression changes over time in an organism. An example would be during embryological development when genes are turned on and off to trigger development of certain structures. Some genes are never expressed at all in an organism. An example would be that males don't express genes associated with female characteristics on the X chromosome despite the fact that they have an X chromosome. So how is it possible to control turning on and turning off of gene expression? One way is DNA availability, meaning that genes in highly condensed regions of the chromosome are not accessible for transcription. Keep in mind that in order for transcription to take place, DNA has to be unwound and pulled apart for RNA polymerase to gain access to the DNA. Therefore, if the DNA remains highly condensed, genes in that region will never be expressed. Another method of regulation is transcription factors. These are proteins that bind to control regions near gene promoters to either turn on or turn off gene transcription. DNA is wound around histone proteins to yield chromatin to allow 6 feet worth of DNA to fit inside human cells. When these DNA protein complexes are spaced closely together, as seen on the top, transcription factors cannot bind and gene expression is turned off. When these complexes are spaced further apart, as you see on the bottom, the DNA is exposed and transcription factors can bind, allowing gene expression to occur. Modifications to the histones and the DNA affect how these complexes are spaced. A mutation is the change in the DNA sequence. Mutations can be good, bad, or neutral. A good mutation would be one that improves an individual's ability to survive and therefore to reproduce. This forms the basis for natural selection, which is discussed in the evolution unit later in this semester. A bad mutation is one that causes a disease such as sickle cell anemia or cancer, while a neutral mutation has no effect on survival. An example of a neutral mutation would be eye color. So how do mutations change nucleotide sequences? There are several different types to discuss. We start with a point mutation, which is a change in one to a few nucleotides in a gene. A specific type of a point mutation is a substitution in which one nucleotide is replaced or substituted with another. An insertion involves adding nucleotides into a gene, while a deletion removes nucleotides from a gene. Finally, a frame shift mutation shifts the reading frame of codons by adding or deleting nucleotides in a multiple other than 3. Recall that codons are triplets. So if a codon is added or removed, that is the only codon that changes. If nucleotides are added or deleted in a multiple other than 3, this would change every single codon after the insertion or the deletion. The deletion of 2 nucleotides, TA in the second codon in this case, shifts the reading frame of an mRNA and changes the entire protein message, creating a non-functional protein. The codons are boxed in red, and you can see that each codon, starting with the second one, changes, resulting in each amino acid changing in the protein that will be translated from the mRNA as well. We just discussed mutations in DNA, but how do the mutations in genes affect the proteins that they code? Well, first, the protein may actually stay unchanged because a codon could be changed that encodes for the same amino acid so that the amino acid does not change. A second scenario is that the new protein may be functionally equivalent to the original one, meaning that an amino acid is changed, but it does not have an effect on the overall function of the protein. The figure on the left shows the genetic code for translating each nucleotide triplet or codon in mRNA into an amino acid. Since the amino acid leucine, for instance, is encoded by 6 different codons, a mutation that changes one leucine codon to another would still yield the leucine amino acid in the protein. On the right is another example of a so-called silent mutation in which the second codon is changed, but it still codes for the amino acid valine. The first 2 scenarios we discussed did not change the protein structure or function. However, certain mutations in genes do certainly affect the proteins that they code. First of all, protein function may be changed by altering the amino acid sequence. In this case, an amino acid would be changed that's important for protein function. Protein function may be destroyed altogether by inserting a premature stop codon. In this case, a codon that encodes for an amino acid is changed to a stop codon. This would yield a truncated protein, which may not be functional depending on how short it is. Sickle cell anemia is caused by one nucleotide change in DNA that changes an mRNA codon that results in one amino acid changing in the hemoglobin protein that binds to red blood cells to carry oxygen. This one change lessens the ability of red blood cells to carry oxygen, which has detrimental effects on the body. So how do mutations in DNA occur? First, mistakes could be made during DNA replication that are not corrected. We'll see in the next unit that DNA replication needs to take place prior to cell division. Environmental factors can also damage DNA. For instance, toxins found in cigarette smoke or ultraviolet radiation in the sun can cause diseases such as cancer. There's a clear correlation between smoking and lung cancer, or overexposure to the sun and skin cancer. Mutations in DNA are important for many reasons. First, they lead to genetic differences amongst individuals by creating alleles. These are alternative forms of the same gene, which could be as simple as somebody having brown versus blue eyes, but these could also be differences that allow certain members of a population to survive to propagate a species. From a clinical standpoint, bacteria are mutating to become resistant to antibiotics. This means that it's becoming harder to clear bacterial infections from individuals. If you've ever heard of MRSA or methicillin-resistant Staphylococcus aureus, you're aware of this problem. Viruses can also mutate to infect multiple species. For instance, avian bird flu was a virus that started in birds and was able to spread in humans. What about swine flu? That was a virus that started in pigs and also mutated to be able to affect humans. Another important point regarding DNA mutations are homeotic genes. These encode transcription factors that are expressed during embryological development. Changes in these genes have influenced animal evolution. Because if certain genes are not turned on during embryological development, this will result in animals being born with different characteristics, which will change a population over time. An application of DNA mutations is in agriculture, in which plants and animals have been genetically altered to yield desirable characteristics. We'll talk much more about the applications of DNA mutations in the DNA technology unit.